This is the Chapter 32.5 overview on Phylum Ctenophora. Phylum Ctenophora is in Kingdom Metazoa, or Animalia, subkingdom Eumetazoa, which designates animals with true tissues. Phylum Ctenophora are different because they are classified with diploblastic radiodens and experience determinate growth, yet they exhibit a biradial cleavage and have 3 germ layers, yet are caught in between the lines of diploblastic and triploblastic and are considered pseudocoelomates. Species in Phylum Ctenophora are known as comb jellies, sea walnuts, and sea gooseberries. This is a picture of Phylum Ctenophora's comb jelly. As you can see, it has 8 rows of comb-like plates of fused cilia. When looking in the biology textbook in Chapter 32.5 Eumetazoa, Animals with True Tissues, you will find the learning outcomes associated with Phylum Ctenophora. These are the 2 questions presented. Question number 2 asks you to differentiate between cnidarians and ctenophores. The answer is members of the phylum Ctenophora lack nematocytes. They propel themselves using 8 rows of comb-like plates of fused cilia. Ctenophores have cells called coloblasts that assist in prey capture, and the symmetry of the ctenophores is a subject for debate. In question number 3, why is the phylogenetic position of ctenophores uncertain? The answer is found by looking at the key morphological characteristics of the number of body layers ctenophores have, which is 3, yet still only having a pseudocoelom. Does that make an animal diploblastic or triploblastic? And which is the ancestral condition, diplo or triplo? Recent studies have suggested that they are in fact triploblastic, with a synapomorphy or characteristic present in an ancestral species and shared exclusively in more or less modified form as that of bilaterians. In addition to that, ctenophores exhibit a modified type of radial symmetry, which suggests that this character may not link them to cnidaria like formerly thought, and instead as possibly closer to the ancestor of early metazoans and precursor of bilaterians. The phylogenetic position is unclear for Phylum Ctenophora. They were formerly thought to be closely related to cnidarians because of their medusa-like form. However, they lack nematocytes like in cnidaria and are structurally more complex than cnidarians. They have anal pores for water and other substances to exit the body. They have muscle cells derived from mesoderm and should be considered triploblasts. The 2 tentacles on either side of their bodies suggest some bilateria. Possibly at the base of the tree, sister to both sponges and eumetazoa, possibly the first, suggesting the ancestral animal could be triploblastic. Ctenophores show the first development of true tissues. They are spherical or ribbon-like, and most are transparent and only a few centimeters long. Rare ones are intensely pigmented and reach 1 meter in length. They propel themselves through the water with 8 rows of comb-like plates of fused cilia that beat in a coordinated fashion, scattering light so they appear like rainbows. Ctenophores are the largest animal that use cilia for locomotion. Many are bioluminescent, and they have two long retractable tentacles used in prey capture. The epithelium of ctenophore tentacles contains coloblasts. Coloblasts are a type of cell that bursts to discharge a strong adhesive material on contact with animal prey. The comb jellies use cilia for movement. Ctenophora, or comb jellies, is a small phylum of medusa-like animals that propel themselves with bands of fused cilia. They may be triploblastic, and they capture prey with coloblasts, cells that release adhesive.